Thank you for joining this webinar on phages best practices for public health. I'm really stoked today to introduce Kevin to give us an overview of the sort of best practices for public health bioinformatics pipelines. I'm just going to go through some sort of like ground rules as to what to expect before I introduce Kevin and let him do his thing. So I'm sure everybody on the call will be buzzing with questions for Kevin. And if you are, then please do pop them in the chat on Zoom. Alternatively, I think there's a system on YouTube where you can also add comments and they'll get fed through as well. The idea being that we will be asking Kevin questions after he has finished his talk. And I guess it'll be on like a first come, first served basis. But if you'd like to get in contact with Kevin to get your question answered, I'm sure he'd be more than happy to receive correspondence for that. And yeah, I think that's pretty much it for ground rules. So I will introduce Kevin to you. Kevin is a bioinformatics scientist who's dedicated his career to addressing the challenges of integrating DNA sequencing and bioinformatics technologies that strengthen public health decision making. And through his advocacy for collaborative and open source bioinformatics development, he's become a key figure in public health bioinformatics. He co-founded the State Public Health Bioinformatics, or STEP-B consortium, and recently served as vice chair of the Public Health Alliance for Genomic Epidemiology's bioinformatics working group, that is Theiagen. So as the Chief Technology Officer of Theiagen Genomics, his role is to advance bioinformatics integration for our public health partners. And the work there has led to the adoption of Theiagen's open source solutions by over 40 US states and local public health laboratories, as well as dozens of academic and government institutions across the global public health community. And Kevin's success really in these initiatives stems from his ability to communicate directly with public health scientists, myself included actually, facing critical challenges and allowing these conversations and relationships to guide Theiagen's software and training development priorities. So with all that in mind, Kevin, if you are ready to make a start, to have your presentation ready, then we'd be really stoked to hear it. So take it away.
Yeah, absolutely. Thanks, Amy. For folks on the call, this is actually a pre-recorded talk. So I think Jamie's going to kick this off. We'll all watch my recording, and then we'll have about 20 or so minutes at the end of this for questions and answers and discussions and things like that. So Jamie, cue up the video. Nice.
All right, welcome everyone to today's Phage webinar. First off, huge thanks to Phage for giving me a bit of a platform today to speak on a project that I hope is helpful to many public health laboratories and bioinformatics scientists in the audience today. So for those of you who don't know me, my name is Kevin Libuit, and I'm from Theiagen Genomics. And I'm also an active member of Phage. And so within Phage, I help to lead a small group within the larger pipelines and visualizations working group. And so over the past couple of months, our sub-working group has been working on a guidance document for the broader public health community. And this document is really just our best attempt to highlight best practices that have emerged in our field over the past decade or so for developing bioinformatics pipelines meant for applications in public health bioinformatics. So today, I'll be speaking about this guidance document in an effort to, one, spread awareness of the work we're doing here, but also, and maybe more importantly, seek community feedback from other bioinformatics developers on this subject. As I said, I represent the work of a small sub-working group, but to get buy-in for this kind of a guidance document, we really need a wide range of practitioners weighing in on what we put together so that the end product is a more proper reflection of the best practices that are being established in our field. So really, what we've aimed to do here is at least provide some kind of starting point for an ongoing discussion. All right, so with that little intro, let's hop into things. So let's start out by first talking about public health bioinformatics pipelines. Pipelines, sometimes referred to as workflows, are a specific type of software that string together a sequence of distinct analytical steps. And in recent years, bioinformatics pipelines have become increasingly critical for public health pathogen genomics, as these software help to make sense of the large volumes of sequencing data that laboratories all over the world are spending millions of dollars to generate. So being able to rapidly adopt bioinformatics pipelines for routine analysis is foundational for public health laboratories. But this process of implementing bioinformatics pipelines has been a huge challenge for laboratories. And this challenge is really further exacerbated by the fact that so few pipeline development practices have been properly articulated in our field. This leads to a lot of inconsistencies in how pipelines are being developed. And we've certainly matured as a field, especially over the last 10 years or so, but without capturing and highlighting what we know are best practices for pipeline development, we leave ourselves susceptible to avoidable issues regarding accessibility, reproducibility, and interoperability. And I think Lee Katz and the folks at the MicroBinFeed podcast capture this sentiment best when they say that there's so much information in the field, but no one really writes it down. And so this is the challenge our sub-working group cared to take on, writing down the best practices in bioinformatics pipeline development that have emerged over the past few years. And as I said, we're a small cohort of public health scientists representing various parts of the world. And within our sub-working group, we have an expertise in both writing and deploying bioinformatics pipelines for public health pathogen genomics. And as we've worked towards this project, our guiding light has been to create a living document of best practices for public health bioinformatics pipelines. And again, I want to emphasize the living document part of this. More than anything, we are hoping to start a conversation around these best practices. And we really invite folks to take a look at what we proposed and provide their honest feedback for us. More voices and perspectives here only help to strengthen our efforts. And it's also worth noting that our sub-working group has been inspired by other similar efforts in the field. Most notably, Torsten Siemens' 10 recommendations for creating usable bioinformatics command line software, and also the work being done by the Journal of Open Source Software, or JOSS. Simply put, we're taking the ideas of these two works and applying them specifically to bioinformatics pipelines for public health. Hence, the title of our guidance document, 10 Best Practices for Public Health Bioinformatics Pipelines. Our 10 best practices offer a framework designed to guide the development, testing, and maintenance of bioinformatics pipelines. All of these practices help to enhance pipeline accessibility, reproducibility, and interoperability, also sustainability of these critical resources. And similar to the JOSS framework, we've written simple yes/no questions for each best practice. Yes being an indicator that the pipeline is adhering to a best practice, and no being an indicator that it is not. Within the document, we've also included guidance on how to answer these questions as a means to assess adherence to each practice. And we've made all of this available through a publicly accessible GitHub repo. So you can check things out now at github.com/phage/public health pipeline best practices. And so here are the 10 best practices in the yes/no questions I mentioned before to gauge adherence to each practice. And at a quick glance, hopefully to most on the call, these are relatively non-controversial practices. As I said, this is more than anything an attempt to simply write down what has been learned over the past few years in our field. But if you feel that we're either missing something crucial or maybe that one of these practices is unreasonable for public health bioinformatics pipelines, you can share feedback directly to our group through that same GitHub repository. So for the rest of my talk today, I'll be diving into each of these to provide a bit more context on these practices and their impact on public health bioinformatics pipelines. All right, so starting with number 1, publicly accessible repository. Is the source code for this pipeline available at a publicly accessible repository URL? As I said at the top of my call, simply accessing bioinformatics pipelines is a challenge for many laboratories. So ensuring that your pipeline is made available through a public repo helps to lower this barrier to access. And today, there are so many free-to-use platforms that can host bioinformatics pipelines. So if you're writing a pipeline that's meant for broad adoption in public health, there's really no excuse not to have your source code hosted on something like GitHub or Bitbucket. So to adhere to this practice, host your pipeline on a publicly accessible repository platform. Simple enough. All right, and number 2, open source license. Does the repository contain a plain text license file with the contents of an OSI-approved software license? Building off the first practice, simply hosting your source code on a repo isn't really enough. Open source pipelines should also include an explicit open source license to clarify permissions for other developers. Including a license within your repo also helps to protect your rights as the original author. Because without an explicit license, users are forced to guess or assume the ways in which they can use your pipeline and maybe modify it for improved functionality. And this level of ambiguity, especially with respect to legal rights of open source software, can be a bit of a deterrent to potential contributors and users. So, to adhere to this practice, simply choose an open source license and host that within your repository. And there are a bunch of resources available that go deeper into this specific topic. One article our sub-working group recommends specifically is the ARS Technica article, Open Source Licenses, What, Which, and Why. This is a great read for folks that would like to know more about this particular topic. All right, and number 3, semantic versioning of stable releases. Are stable releases that follow semantic versioning of the pipeline available for implementation in public health laboratories? For those unfamiliar with this concept, semantic versioning is a versioning system that uses 3 numbers to indicate software versions. These 3 numbers indicate a meaning about the types of changes made to a software from version to version. The 3 numbers represent either major, minor, or patch updates to the code. And use of semantic versioning is so wildly helpful as it ensures clarity and predictability in version updates, all of which helps to facilitate the maintenance of stable, compatible, and reliable tools critical for accurate and timely pathogen genomics. Without clear versioning, a lot of challenges are introduced, especially as it relates to compatibility with other software and keeping up with the most up-to-date versions of a particular pipeline. And adherence to this practice is simple. Implement semantic versioning. Again, hopefully a relatively uncontroversial take on pipeline development practices. Okay, moving on to number 4, workflow management systems. Does the pipeline utilize a workflow management system for its development and execution? And actually, of the practices we've covered so far, this might get the most pushback, as I do know, personally even, many pipeline developers that haven't adopted workflow management systems. That said, as a sub-working group, we really wanted to include this as a best practice because the utility in workflow management systems at this point is really undeniable. For those unaware, workflow management systems, for example, Nextflow, Whittle, or even SnakeMake, have dramatically standardized the process of pipeline development. Rather than a home-brewed custom attempt to string analytical tools together, workflow management systems offer an out-of-the-box framework to do so. And these frameworks are often inherently compatible with various compute backends, meaning pipelines written in these systems can be readily deployed in cloud environments, on a local machine, or even within a complex HPC system seamlessly, all of which enhances the portability of these pipelines, as well as increasing the ability for others to collaborate with one another. And so in the spirit of a best practice, this really has become the status quo for pipeline developers. Because without the use of a workflow management system, developers may unnecessarily introduce challenges to implementation, especially if end users are working in different types of environments than the author originally envisioned. And so to adhere to this practice, simply use a workflow management system for your pipeline. And we'd recommend one that has been proven in the field. For public health pathogen genomics, I'd recommend the use of things like Nextflow, Whittle, or Galaxy. SnakeMake is also a great option for folks that are more comfortable with the Python language. All right, and number 5, containerized/packaged software. Does the pipeline utilize containerized software, for example, Docker, or packaging, for example, Conda, to enhance interoperable pipeline distribution? Containerized software and packaging have been critical technologies that help to promote portability of analytical modules within the pipelines themselves. Before the wide adoption of Docker or Conda, getting software installed on a system took a ton of technical expertise and was so susceptible to errors, meaning a pipeline installed on one machine could end up generating different results from that same pipeline being installed on a different machine. So adopting the use of containers or packaged software in pipeline development has become a best practice, as it helps to avoid all of these challenges with software dependencies. And ultimately, this is a best practice that helps to promote reproducibility across public health labs. So to adhere to this practice, rather than dependence of local installs of each analytical module, take advantage of software containers or packaging. For containerized software specifically, I'd recommend checking out the StaffB Docker Builds project. This project hosts software containers for hundreds of bioinformatics tools and is actively maintained by public health scientists in the US. Worth noting, a bit of bias here, as I am currently an admin of this project. Nonetheless, it's a fantastic resource for the public health community, evidenced by the millions of pools of R containers in laboratories across the world. All right, and moving on to number 6, common file formats. Does the pipeline accept as inputs and generate as outputs common file formats utilized in public health pathogen genomics? For things like read data, variant calling, phylogenetic trees, annotation files, and really all other information relevant to pathogen genomics, there exists a common file format to store this data. And the best practice is to adopt that file format for your pipeline. And if there is no file format that exists for the type of data you're storing, it's likely an indicator that what you're working on may be more of an academic tool that may need more scrutiny across the field rather than a pipeline meant to inform public health decision-making. And so with the use of common file formats, again, you're promoting interoperability. This also helps to ease data sharing and even integration with other pipelines. When a pipeline opts to use an uncommon file format or even a custom or novel file format, it really just adds friction to data exchange. Often, this requires other developers to write a tool that converts the uncommon file format into a common file format. So to adhere to this practice, dive a bit into the research to identify the appropriate file format for the data you're storing and adhere to that formatting. It will go a long way towards interoperability. All right, and number 7, software testing. Are there automated and or manual tests described so that the functionality of the pipeline can be assessed? This is another one that I know a lot of pipeline developers may not necessarily be practicing right now. That said, again, similar to workflow management systems, our subworking group felt strongly in including this as a best practice, as it really should be the status quo. When writing a bioinformatics pipeline, some system of tests should be implemented to continuously assess the functionality of that pipeline. Now, bugs and errors will always arise in software development, but the inclusion of tests in your development practice helps to at least add some kind of mechanism to filter bugs that may lead to consequential workflow errors. And by creating these types of tests in your software, it helps to provide confidence to your user base that precautions against these unintended changes are in place for your code. Because without testing, issues and bugs can go easily undetected. And so to adhere to this practice, start writing tests in your pipeline and provide some description of the tests being done to ensure reliability and accuracy. And if you're a developer and you're interested in learning more about this subject, I can't recommend highly enough to check out the ASM NGS 2020 hackathon available on GitHub. This was a virtual hackathon hosted by folks within the micro-BinFi community, and it's an amazing resource to learn more about software testing, both its importance as well as how to use things like GitHub Actions to set up automated testing of your pipeline. All right, and number 8, benchmark/validation datasets. Is there a publicly available set of inputs with known outputs that can be used to test successful installation and benchmark against other tools? These kinds of data sets can be huge for users of your pipeline, as it allows them to give an assessment of proper functionality on their end. Even if it's just a small data set, this can go a long way for users that may be working with your pipeline for the first time. Because without any sort of benchmark or validation data sets with known outputs, the effectiveness of the pipeline itself, especially to new users, may be unclear. So, to adhere to this practice, include some kind of benchmark or validation data set along with your pipeline. And this doesn't necessarily need to be a large data set of raw data that's hosted somewhere. Often, making a list of INSDC accessions will suffice, at least for pipelines meant to analyze FASTQ read data. And number 9, also a practice related to data sets, reference data requirements. Are required reference data and/or databases clearly documented, publicly accessible, and maintained? This is another critical practice that we wanted to include in this document because so many pipelines nowadays rely on external databases for analysis. SARS-CoV-2 analytical pipelines are a great example of this. If your pipeline is performing SARS-CoV-2 lineage assignment, it likely depends on a database maintained by the Penguin Network. So letting users know that this is the case is critical, especially if your pipeline requires a specific database version to run properly. Without including this type of information, issues with reproducibility can be introduced as users may not even have access to the required references for your pipeline to run properly. So adherence to this practice means indicating what references are required and ensuring that these reference files are, similar to the pipeline itself, publicly accessible and maintained. And if external references are required, those datasets should also adhere to static versioning, open access, and clear instructions for installation and access. And number 10, last but certainly not least, pipeline documentation. Is the pipeline documentation clearly written and publicly accessible? This is a huge one and absolutely essential for public health pipelines. Without proper documentations, users will have to go through painstaking efforts to really understand what's happening under the hood. And this practice of pipeline documentation is really more of a categorical practice because there's so many components to strong documentation. So to properly adhere to this practice, we've included several document practices, all of which should be followed to adhere to this practice of pipeline documentation. And so here are the pipeline practices we've communicated in our guidance document. And most of these are really straightforward and can be applied to really any sort of open source software, such as contribution, authorship, and verified point of contact, and conflict of interest statement. And we don't necessarily have the time today to get into each of these, but I did want to take a second to highlight one documentation practice that we have had the most debate about, both internally and externally. And that is 10C, Pipeline Maintenance Statement. So let's look at this documentation practice, Pipeline Maintenance Statement. Have the authors provided documentation regarding the intent to maintain the pipeline? Including this kind of information can be enormously valuable for public health laboratories, especially when you consider that major public health decisions may depend on the pipeline itself. So some kind of communication to users that there is an intent to maintain this pipeline is very useful for public health labs. And in some instances, this statement may indicate that the developer themselves don't actually intend to maintain the pipeline. Instead, maybe it's coming from someone who may be on their way out of a laboratory, but perhaps the author wrote the tool in a way that would instead encourage some kind of community maintenance. For example, if the original author wrote the pipeline using a standard workflow management system like Nextflow or Whittle, this would dramatically reduce the burden of maintenance for the original author. And instead, it could be a tool that is maintained by the broader developer community. Either way, including in your documentation some kind of statement regarding the maintainability of the pipeline itself can be incredibly valuable. So to adhere to this documentation practice, simply include some kind of statement about the upkeep of the pipeline. And I wanted to take some time during today's presentation to talk specifically about this documentation practice, because unlike the other practices in this document, this is really one that we don't see much of in the field. Instead, it's more of a new practice that we as a sub-working group wanted to propose to the community, given how critical sustainability is in public health pathogen genomics. All right, so here they are again, all 10 best practices for public health bioinformatics pipelines. Hopefully this was helpful to folks on the call to learn more about the perspectives from our sub-working group regarding this topic. We also hope this was helpful for the developers who may be writing their own pipelines. We hope this document provides a framework for you as you build your tools that are meant for adoption across public health. In the end, all of this can be found through our GitHub repo, github.com/phage/public health pipeline best practices. And so this is where we are as a sub-working group now. We've recently published these 10 best practices on our repo. So as far as the next steps, the most immediate one is seeking community feedback. And that was a big motivator for me speaking on this webinar today. This is an opportunity for us to communicate our work to the field so that we can hear others' thoughts on the matter. And today, I know we'll have plenty of time for questions and discussions. So if you have any thoughts of any of the practices we've included in our guidance document, we'd love to have this conversation with you. And I know that there are many others from our sub-working group on the call who may be able to also add their insights on your questions or critiques. And so beyond community feedback, we're also hoping to move forward to create resources that help promote the adoption of these practices. One idea we have is creating some kind of JOS-like checklists on GitHub. This would serve as a resource for developers to quickly assess their own pipelines to see if they're adhering to each practice. And this kind of resource may also serve as a mechanism to communicate to others, both developers as well as users, that pipeline developers have chosen to adhere to these practices. And from there, we're also looking into ways for us to highlight software that do adhere to these practices. This might be something we could accomplish with GitHub badges and may be most relevant to pipeline users within public health laboratories. For example, when a lab is looking for a pipeline that serves a specific function, it can be a bit daunting when sifting through the many pipelines made available on the internet. So we're thinking that something like a badge system or even a simple list of pipelines that meet these standards can help narrow that search space for public health laboratories. The projects listed here are still very much in the nascent stages of development, but this is more or less the direction we see ourselves walking as a sub-working group. All right, and so that's it for my talk today. I again want to emphasize the incredible collaborative nature to this work. This has been a big effort across not only our sub-working group, but really the pipeline and visualizations working group at large. So huge thanks to the chairs of our working group in Emily Smith, Gultiken Unal, and Victoria Carr. As a previous working group chair, I can certainly appreciate the effort it takes to running a working group. And these three have really done an incredible job in helping to keep us organized and moving forward. And I've also included the list of folks within our sub-working group that have contributed directly to this document. Huge thanks to everyone for their incredible efforts on this project. One last thing, if you're at all interested in contributing to this kind of work, please feel free to reach out and we'll be sure to loop you in to future conversations. So with that, thanks again to the phage organizers for giving me this opportunity to speak today. And now I'll be happy to take questions from the audience.
I'm not hearing your audio, Amy, though. I don't know if that's just me. I'm not hearing anything. All right, as Amy figures out some of the audio things, I can go ahead and look at some of the questions in the chat. Thanks to everyone there. And there was a couple I was able to answer while going through things. So I can rehash some of those questions from, it looks like Newton. I apologize if I'm mispronouncing that. The first question in the chat I was able to answer while the recording was going is a pretty funny question about the community name of phage. Newton asked if we all, because of the naming coincidence, if there's any focus on bacteriophages. And the answer is oddly enough, no, but it's certainly a coincidence we're all well aware of, but there is no, at least to my understanding, any focus of bacteriophages across the phage organization. Newton also asked a couple other questions, one of which inquiring about tuberculosis software for public health settings. And then I also included, as an answer to that, some of the workflows we've made available at Theiagen, namely Theiaprobe, which was written for analyzing bacterial data in public health settings. And there's a specific module on TB that takes advantage of Jody Phelan's TB profiling. So Newton, hopefully that's something you find helpful there as well. And then in terms of a soft copy for the documentation, I've added the GitHub link to where we're hosting this. It's within the phage organization. You should be able to just go to / public health pipeline best practices to get to that, the copy of the document that I was walking through today. So hopefully that answers your questions there. All right. And going through these other questions, I'm seeing one from Alnesh. Just giving us a nice thing. Appreciate that. Calling in from Ethiopia. I see a question from Hector Ruiz. You have recommendations of resources to learn and teach use of workflow management and containers. Yes, there's a couple of different things I can mention. One of which at Theiagen Genomics, we've hosted a couple of these trainings and made those resources available on GitHub. So let me find those links and I can throw that in the chat there as well. So the first link I just sent you is a workflow management course that we hosted for laboratories in the US. So in there, you'll see exercises, lectures that focus on specifically Nextflow and the workflow development language WDL. I think the NF4 folks and the folks at Nextflow and Takara, they have a number of different resources for training as well, if you just Google something on that. And in terms of Dockers, I'm also going to throw another training that we hosted here. This was something we did in collaboration with laboratories in the Northeast of the US as well, and the SAFE Docker Build repository. You should be able to see some of our, again, exercises, recordings, lecture content, and everything else there. So Hector, yeah, hopefully those are resources you find valuable then. All right, I see another question from Valerie. Just to find out how you intend to look at things like automation, reproducibility, and hopefully scalability and resource efficiency. Oh, that's a fantastic question. And I'm guessing you're talking about in terms of just the high volume of data being generated. There's just a need for automation of these kinds of workflows to be running and ensuring that reproducibility. So really, I would say all of these practices kind of help to address this part of the conversation, especially with respect to reproducibility.
And specifically on the conversation of scalability, I'd say that's where the workflow management systems come into play. Like I mentioned in the talk itself, languages like Nextflow, Whittle, SnakeMake, and all the rest of those workflow management systems, they've been written with scalability in mind. So at the first pass of things, you'll notice whenever you write software in these workflow management systems, there's an inherent parallelization of samples that are getting analyzed. So you can define the compute resources per sample, and whenever you launch analysis on a data set, the compute resources are separated by sample. And then adding to that is their inherent compatibility with things like cloud infrastructure. So whenever you're using something again like Nextflow, there's an inherent compatibility with configurations on AWS batch, GCP, whatever GCP batch system is available there as well. So there's a paved route, I would say, even to scalability using things like Nextflow, Whittle, and all the rest. Automation is probably a different type of question beyond the scope of necessarily the document we were talking about, because really what our narrow focus was in putting this guidance document together is on the pipeline conversation. And we had to whittle that down, no pun intended, over the couple of months we've been working on this document, because there's so many aspects of pipeline integration that we're talking about, including automation, like you had mentioned. So with respect to this guidance document, automation wasn't necessarily a component of what we were trying to address, but nonetheless, it's still a major factor to consider when you're looking at public health settings, and it's likely throughout the working group at large, questions on automation are going to be addressed. And even not even just the working group, but other working groups, like this might be something the infrastructure folks may also take on as well. So Valerie, hopefully that gives you some perspective on both the automation, reproducibility, and scalability of these resources. All right, and I see a question from Kofi Hongo. I prepared a small course in bioinformatics workflows for your PhD community. Could you provide some useful tools? Yeah, absolutely. If you look back in the answered questions in this chat, you should hopefully see some resources we put out there for Hector's question. Again, that was a course that our team at Theiagen helped to put together and host. Other things I would recommend in terms of Nextflow, as I mentioned, Theiagen and their whole group have put a number of different live webinars and recordings that are publicly accessible. On the Whittle side, I'd be remiss if I didn't mention Lynn Langit. She has a whole YouTube series on workflow management and specifically Whittle, use of mini Whittle. So I can see if I can find her resources for you now. Yes, all right, we got it. And Kofi, hopefully this will be something you find value in. All right, solid. So Kofi, there is a link to also Lynn Langit's series on Whittle. Incredible resource that she's put together there. All right, and I see an anonymous question here. What is your personal opinion on Nextflow? Even though it's supposed to make things easier, sometimes debugging, troubleshooting is way harder than on homebrew code, at least in my personal experience. That's fair. Whomever that anonymous writer is of that question. In terms of my personal opinion, I have a couple things to say on that. Like, Nextflow is an incredible resource, and I definitely understand that that transition state maybe you're going through in terms of difficulties in debugging and troubleshooting. What I would say is it's not necessarily harder. I would advocate in changing that perspective that at current, it may just be something where you're learning something new. Whether or not it's harder or easier, I'd imagine if you have collaborators, as an example, it's going to be much harder for those collaborators to jump into a homebrew code rather than maybe looking at something that's a bit more standardized like Nextflow. So the whole Nextflow community, of which is hundreds of developers, if not thousands, are working to optimize what's expected for debugging and troubleshooting. So they have really robust systems for logging these types of things. It's just something that will be new to learn as you start to adopt those languages. And in terms of my personal opinion, it's fantastic language. It's very flexible. That's probably something I would highlight, relative to something like Whittle, which I write a lot in. Nextflow is very customizable, flexible language that can meet really the bespoke needs of different practitioners in the field. And yeah, I'd recommend, as part of it is to learn how to do the debugging and troubleshooting. It's worth learning, in my opinion, because as I mentioned through the talk, it's becoming a best practice to use these languages, to use these systems and frameworks to create these pipelines. And if you do have issues, there's a whole community of practitioners, especially for Nextflow, that are more than willing to help you out and walk you through that process of debugging and troubleshooting and all the like. So hopefully that gave you some perspective there as well. Ooh, did I get that one in? All right. There's a question from Yursa. Can you tell me about the access of NF4's Viper pipeline packages? I'm trying to install them, but they're not showing availability. Usara, I'm sorry if I'm mispronouncing my name. Unfortunately, I'm not directly tied to NF4's Viper pipeline package, so I don't have any kind of control on the access issues there. But I'd recommend, I'd imagine if there's GitHub repo or something of the sort, reaching out directly to those authors, and hopefully they'll be able to walk you through that access channel. All right, and I see a question from Simon. Are Phage planning to provide a list of recommended file formats, similar to for benchmarking validation for common sense? Yes, exactly that, Simon. So as soon as we put that out there as a best practice, that was one of the immediate feedbacks we got both internally and externally. When we mentioned the common file formats, how we should be able to list some kind of reference document indicating what are the common file formats for the various types of data stored or accessed and shared across public health pathogens and genomics. And actually, that was one of the guiding, or maybe we'll call it an impetus for creating a separate repository for this guidance document specifically, is we knew that there was going to be some auxiliary resources that we needed to create to coincide with the guidance document itself. So one of which being a list of those common file formats and their purposes in the field of public health pathogens. So great question, Simon. All right, walking through a couple of these. Tracy, I see your question that Phage also has a tuberculosis pipe biz working group on pipe. Yes, on pipeline. Newton is welcome to join us. Thank you, Tracy, for mentioning that. So yeah, that was a question I think Newton asked previously. So just adding some context there that within the whole pipe biz working group, as I mentioned, I'm representing a small sub-working group. There exists another sub-working group that's specifically addressing questions and concerns regarding tuberculosis in public health settings. In Newton, that might be something you'd be interested in joining. All right, Anonymous Praise, thanks for all that. Appreciate this. So working through. All right, and then there's a question here about provenance. So it says, thanks for the talk, Kevin. What recommendations do you have for provenance? What type of information to capture in this document? Oh, that's a fantastic question. Maybe a bit outside of the scope of this guidance document specifically, but conversations with data provenance, where data is coming from, what's happening to it along the way, and how to actually format that is an ongoing conversation. I think maybe a little bit outside of the context of this, but something for sure across the Python visualizations working group will be addressing moving forward. So thanks for that question. We'll be putting more information about that, I'm sure, in future working group conversations. All right, and working through these as fast as I can, so apologies if I'm speaking quickly. I just see quite a bit of questions here. All right, Alfred, thanks for these very important insights. The guidelines are really great for labs with some good bioinformatics capacity. Appreciate that. So Alfred is asking, any thoughts about best practices to create tools that can be used at labs where there is not much bioinformatics capacity? For example, for remote facilities in low resource settings with intermittent access to internet and similar challenges. Oh, that's a fantastic question, Alfred. And I think the biggest challenges in what you're speaking about is going to be that the intermittent access to the internet. And I think that's probably, there's this conversation of what, where things need to be addressed first in terms of capacity development. And it is really difficult to do bioinformatics without internet access. It is obviously the caveat situations where you're going to need remote capabilities. It's just that at present, the solutions that fit those remote settings require pretty high technical capabilities. Predominantly, I'm speaking of like command line interface, know-how, and awareness. Outside of maybe proprietary software, like things that you could probably bring on site, like CLC Workbench or Genius and all the like. Outside of that, if you're talking about open source software that doesn't rely on the internet, you're going to need a very highly technical individual that's willing to go out there and be able to provision those things. That or someone with that kind of access. So if the internet challenge is addressed, then your options open up dramatically. One platform, especially in resources where they don't necessarily have the bioinformatics technical capabilities, we found favor in is things like the Terra platform that's developed and maintained by the Broad Institute of MIT and Harvard. We found that to be an incredible resource for public health laboratories. Other things like CZ Gen Epi, really all the resources coming out of the Chan Zuckerberg initiative in BioHub, CZID being another one that's more focused on metagenomics. Galaxy Project, I think we mentioned it here as well, also another graphic user interface to get access to these open source tools. But all of those three platforms require internet access. So Alfred, if you want to keep talking about this, I would also point you to the Data Infrastructures Working Group at SPADE as well. They can give you some more insights there. All right, working through, I think I got 5 more minutes. I'm going to try to work through these as quickly as I can. So do you have a training module on pipeline writing? Yes, we do. I threw that as an answer in a couple of previous questions. Seeing something from David. Do you have a thought on reporting pipeline outputs to clinical or epidemiological colleagues in the context of outbreak characterization? For example, do you recommend emailing a phylogenetic tree or providing written narrative or something else? Thanks for the talk, David. Fantastic question. And what we're seeing as a practice kind of emerging, a best practice, if you will, emerging in this is even before deciding what goes into those reports is the educational discussions with your epidemiologists, because they themselves might develop a preference between, hey, I'd like a SNP tree or just like a SNP matrix, or maybe it's just, hey, I just need the accessions of the isolates that have been identified within that putative cluster. So I think before starting necessarily with that endpoint in mind, I think the endpoint is a collaborative discussion or collaborative relationship rather between the laboratories and the EPIs. And I would start first with that educational conversation and perspective, so then you can build that report to the needs and preferences to your endpoint stakeholder there. All right, Tim Dolman, Kevin and colleague, great work on this much needed effort. I appreciate that. I wonder if you can speak some more about the use of specific workflow repos, NF4, workflow hub, etc., and how they fit into this work. Oh, Tim, I don't know if you want to add a little more context in what you're asking specifically about those workflow repos. I guess I can give a perspective just in the realizing I have just a bit of time here left, but things like NF4 are amazing. Workflow hub and all the like, these repositories where you're seeing reusable modules that get incorporated into different pipelines, again, further emphasizing the interoperability and reproducibility of these analytical modules that get brought into these bioinformatics pipelines. So those further kind of aim to standardize our field that has, historically been just described as the wild, wild west. Now you're seeing, common code types that are being brought into different pipelines and laboratories, and I think that's only a reflection of the maturation of the field. You don't have to reinvent the wheel every single time. Kofi, another question. Is there community help to fix common errors in workflow systems, or can you just DM us sometime? So there's generally going to be some kind of technical working group associated with the workflow language you might be adopting. Unfortunately, I would not recommend DMing me specifically for every workflow error. It's likely going to get lost in the inbox. But I would recommend maybe looking at things like the phage working group, so you can be put in contact with a group of other technical practitioners. Another resource I mentioned in the talk was the MicroBinFi podcast. So feel free to use that as well, Kofi, especially as you're learning through these things. Adriana, do you have recommendations on best practice guides for database curation and maintenance? I do not, but that's a fantastic question. We have a data structures working group that has immense experience in data. There's a lot of database curation expertise on there, actually. So I'd recommend checking out the other phage working groups on this topic, Adriana, to get perspective there. All right, this is probably the last question. I apologize. I was working as quickly as I could through these. But there's a question: how do I stay up to date with tools currently being used in bioinformatics analysis? And how do you determine what the best options are? And I would say the way in which we're currently doing that is being a part of phage, having that ongoing technical group where we can discuss the happenings in our field, being a part of technical forums like MicroBinFi, their MicroBinFi podcast. I also run a podcast with Andrew Page called the Bioinformatics Lab Podcast, where we try to communicate these different things as well. All right. Sorry again for talking as quickly as I can. I'm getting notes from the host about time being up in 1 minute. So for folks that are interested in continuing on this conversation, again, I'd recommend checking out the phage website and resources, and you can get in contact with the Secretariat if you're at all interested in joining our working group and things like this. If you're interested in the work we're doing, again, we posted our phage guidance document, GitHub repo, and you can communicate with us directly through that. One other housekeeping note that this recording will be made available shortly after this on the phage YouTube channel. Sorry again for folks that I didn't get to your questions, but yeah, appreciate everyone taking the time and listening to this talk.
Thank you very much, Kevin. Hope you can hear me again now. Yes, that was a fantastic presentation, and the questions that came in were all outstanding as well. So I'd like to say, obviously, big thanks to Kevin for giving his time to go through this webinar with everybody today. I'd like to thank Jamie at phage for doing the technical monitoring for today. Everybody in the phage working group who has contributed to this project. And thanks to all of you for tuning in to watch this today. I do hope it's been useful for people who are interested in understanding how we're trying to standardize, quite a cutting edge field of science. So just to reiterate, if you'd like to find out more information about joining this wonderful group of people, you can join the pipe visualization working group, and other groups are available as well on our website, phage.org. That is PHA4GE.org. So with that all in mind, Kevin is also available to contact with specific questions if you want more detail, more context. He can be more than happy to do so. Kevin, if you could provide a contact detail, I'll be able to facilitate that at some point. But yeah, I'd like to thank everybody for being here today, and I hope you all have good mornings, afternoons, evenings, depending on where you are in the world.
All right, thank you.